Welcome everybody. Kimberly is very happy that the food is all eaten, so thank you for your we have Professor Colin Nichols today, and Colin is the Cori Professor in Cell Biology and Physiology. Colin received his undergraduate and Ph.D., correct me if I'm wrong, but I think the University of Leeds in England, right?
Correct.
When Mark Boyd was still there.
Correct.
And after that, he went to the University of Maryland, where he did a postdoctoral fellowship with John Lederer. Colin has made tremendous contributions to the field of ion channel biology. Important contributions are the inward rectifier currents, potassium channels, the biology of those channels, gating of those channels, and then of course the KATP channel, that is a channel that links metabolism and electrophysiology, and I think that's what he will talk about today. So, thanks for coming.
Thank you, Colin. That was very generous, and thank you all for showing up. This is a very nice seminar series to speak in, and I'm wondering now when I last spoke, because I'm hoping that I'm not going to tell you something that I already told you 9 months ago. Do you remember when I last did this?
Never.
It's on the website.
Well, anyway, hopefully this will be new to some of you, and hopefully it'll be interesting. So I was trying to, twists in the tail. People were looking at it, what? They didn't get it. Twists in the atrium. And I'm going to talk about things in the atrium. So I'm going to talk about KATP channels, and of course, I don't think I need this slide. The general idea to show you that there's diversity of potassium channel function. KATP channels, when they're turned on, are channels that are essentially non-rectifiers. They're structurally, as I'm going to show you, members of the inward rectifier family, but they really don't rectify significantly in the physiologic range, and they are effectively a linear K-conductance when they're turned on, and so when they are, they will both shorten the action potential as well as stabilizing resting potentials. So the molecular basis of KATP channels, which we've elucidated over the last 15 years, started with the cloning of constituent members, shows that these channels are kind of unique in that they have a pore-forming KIR subunit that generates the pore as a tetramer, just as in all K channels. This is a member of the KIR channel family. It has 2 transmembrane helices, a P-loop, and cytoplasmic amino and carboxy termini. But uniquely, this channel is associated in, as far as we can tell, with biophysical and biochemical analyses, which I won't talk about, a one-to-one, I think this is a one-to-one stoichiometric association with a sulfonylurea receptor, which is a member of the ATP binding cassette family of proteins containing 2 nucleotide binding folds, which when first cloned we assumed, well, this must be where the ATP sensitive channel gets its ATP sensitivity from, these nucleotide binding folds. That turns out not to be the case. I won't go through the data, but we know that ATP inhibits the channel by binding to the KR6 subunit, and what happens at the nucleotide binding folds, at least experimentally, is that ADP binds there, and that activates the channel. This is a cartoon that shows more or less a summary of how we think the channel is regulated at the molecular level. The blue subunits are meant to be the KIR6 subunits. There's a bit of the membrane. This is the business end of the sulfonylurea receptor with the 2 nucleotide binding folds, which actually make 2 nucleotide binding sites. Best as we know is that magnesium ATP is permanently bound at one site, and it's hydrolyzed at the other site, and that magnesium ATP bound state is the active state of the subunit, which acts on the channel to overcome the inhibitory effect of ATP binding at the channel itself to turn the channel on. And if you apply magnesium ATP in an inside-out membrane patch, you can activate channels in the presence of ATP. So that physiologically we think that the primary regulator is the ratio of ATP to ADP. sulfonylurea drugs bind to this sulfonylurea receptor. The sulfonylureas, just to remind you, I don't think it'll come up, but these are drugs that are first-line type 2 diabetic drugs used to block this channel, or rather inhibit this channel, by interacting with this subunit and probably acting to stopping the ADP activatory effect on the channel. So there are two pairs of genes in the eukaryotic chromosomes that encode KATP channel subunits. Chromosome 11p15 in humans has SuR1, and then the immediately downstream gene is KIR6.2. Chromosome 12p12 has SuR2, which comes in two major spliced forms which differ by about the last 100 amino acids of the sulfonylurea receptor. And immediately downstream of that is KIR6.1. So this is very interesting evolutionarily. It means that somewhere in history there was a sulfonylurea receptor and a KIR6 subunit that were expressed together to make a channel, and the fact that they are this is immediately downstream of this, suggests to me that there must be common regulatory elements for expression of these two genes. And then somewhere in history, this whole shebang was duplicated. I don't know whether this came first or this did, but one of them was duplicated to make the other one. But now we find that in native tissues, you can actually have KP channels formed from any pairwise combination of SURs and KIRs, KR6s. So we know that in the pancreas and the hypothalamus, for instance, this combination, SUR1 KR6.2, is predominant and generates the KHP channels. We know that in the vasculature, SUR2B and KR6.1 is a very prominent combination. In the heart, in the ventricle for sure, SUR2A and KR6.2 is a major pair, but I'm going to show you that SUR1 and KR6.2 is also present in the heart. But dogma has it that it's SUR2A plus KR6.2. And I'm not sure where SUR1 KR6.1 is present, but maybe in smooth, there may be some in smooth muscle, there may be in certain brain combinations. But for sure, in recombinant channels, if you express any one of these pairwise combinations, you can generate functional KHP channels. I'm sorry, I've got a cold, and I'm going to dry up and look here. So we know a little bit about the consequences of knocking out the KHP channels in different tissues from mouse knockout studies. And many of the phenotypes that we get from these experiments are exactly as we predict. So loss of KR6.1, which is present in vascular channels, leads to loss of vascular channels. It also leads to a Prince metal angina-like coronary vasospasm. Knockout of KR6.2 leads to loss of pancreatic KATP channels and loss of glucose-stimulated insulin secretion. It also leads to loss of at least ventricular KATP channels, and there is a little bit of evidence that you also lose atrial channels, but we may come back to that point. And you lose ischemic protection of KATP. Knockout of SUR1 leads to loss of pancreatic channels and loss of insulin secretion, glucose-dependent insulin secretion. No reported effects in the heart until, as we published last month, and I'll tell you about, we show that SUR1 is actually critical in the atrium. And knockout of SUR2, which is the purported ventricular isoform, does lead to loss, or at least significant reduction of, that should be ventricular KTP channels. I don't think we'll talk about that. So knockout of KR6.2 or SUR2A causes loss of ventricular channels, and here's the prime evidence. So here are voltage clamp recordings from wild-type mouse ventricular myocytes under control conditions. You see inward IK1 and then outward KV channels. Pinacidil is SUR2 specific KTP channel opener, and it causes a massive opening of KTP channels, which is all blocked by glibenclamide. In the knockout animals, there is no activation of channels by pinacidil. And if we look at the action potentials from mouse ventricle, you see that 100 micromolar pinacidil is enough to shorten the action potential to a spike. There is essentially no duration to the action potential, and it's blockable by glibenclamide, and there is no effects in the knockout animals. And SuR2, this is a point we may come back to talk about. If you look at wild-type animals, wild-type hearts, these are just membrane patches from wild-type ventricular myocytes. You see plenty of KATP channels opening on excised patches, and you see just a few KATP channels opening in SuR2 knockouts. And this is the work of John Mikulski predominantly, and they've been sort of cautious to say over the years that there is not necessarily a complete loss, and they're still at a bit of a loss to explain why that is, but partly it's no doubt because KR6.2 is still being expressed, so the channel isoform is still there. But whether it's associated with SuR1 or whether it's associated with truncated versions of SuR2A, of SuR2, because the knockout is not a complete gene knockout, it's a gene interruption, and so it's possible there are altered spliced forms of truncated versions of the sulfonylurea receptor that are present. So I said I'm giving you a lot of background, so I'm sorry if I'm just trying to educate you for the sake of it, but I think I'd go on about it a little longer to talk a little bit about the physiologic role of KATP channels. How do we envision what happens if we to activate KATP channels? And there's some important numbers to bear in mind. I don't know if I've got the data here to show you, but half inhibition of KATP channels occurs at around 10 micromolar ATP. And there's almost complete inhibition at 1 millimolar if you look at the ATP sensitivity in excised patches. And this has given rise to an age-old question that comes up almost every time I or anybody else gives a talk about the physiology of KATP channels, and that is, well, how can you ever get physiologic activation of these channels when physiologic ATP is here, somewhere in this white bar, in the millimolar range, and yet these channels are all inhibited by 1 millimolar? Well, the way we think it happens is something like this. Let's, for argument's sake, say we start out with an ATP sensitivity somewhere close to 10 millimolar. What happens if you inhibit metabolism? ATP starts to drop. It probably doesn't drop very much, but more importantly, ADP starts to rise, and ADP will activate the channels. And it does so essentially by shifting the ATP sensitivity to the right. So we shift to a new ATP sensitivity curve, and as ATP drops a little further, as metabolism is inhibited more, this will shift to a new curve as MAG-ADP rises. And so even within the millimolar range, what happens, and this is almost certainly what happens in the pancreas, the channels get activated with ATP remaining in the millimolar range because the sensitivity to ATP shifts, and it can be shown to shift this far by adding micromolar, up to hundreds of micromolar MAG-ADP will shift it this far to the right. Okay, so we attempt to address the question over the last 20 years probably of what is the consequences of KATP channel activity, that's supposed to say cardiac, myocytes. And in the last 15 years we've been trying to address the question genetically of what happens if we manipulate KATP to try to deliberately increase KATP. Well, by way of ancient history, this is the prediction of what happens. So on the right are ventricular myocyte action potentials from the rat that I recorded 20 years ago. And it shows the consequences of metabolic inhibition. So if you deliberately inhibit metabolism in the ventricle, and you can do this with global ischemia actually, but if you do it by, in this case, deoxyglucose and cyanide, you see that the action potential shortens, and you can model this effect by just increasing a linear K conductance. So on the left, this is Dennis Noble's Oxsoft heart model, which is a very early progenitor of the sort of models that Yoram has championed. And what I did in this model was to add a linear K conductance, and you see that it shortens the action potential in more or less the same way as we actually observed experimentally. And I scaled the amplitude of the conductance by what it's predicted to be based on the measured ATP and ADP sensitivity of the channel. And so these numbers show what the cytoplasmic ATP is to scale the conductance appropriately. And the main point to notice is that even within the millimolar range, if the ATP starts to drop, this is 16, 8, 4, 2, once you get down below about 4 or 5 millimolar, you start to get substantial action potential shortening. You don't have to get down to low micromolar range. And the prediction of this kind of modeling is that you will get about 50% shortening of the action potential, that would be about somewhere between D and E here, if you activate only 1% of the total KATP conductance. So 1 millimolar is enough. 1 millimolar ATP will actually activate only about 1% of the KATP, but the density of the channels is so high. I showed you at the beginning, if you put phenazole on, you get a huge conductance. You only actually have to activate 1% to shorten the exponential duration by 50%. Now, I said this 20 years ago, and maybe it wasn't likely, but far greater men than I have since said it. And so many people who have since done modeling and experimental studies have come up to the same conclusion, basically, that the KTP density is so high that if you activate 1% of it, you'll get 50% shortening of the action potential. So what this means is that the cells are sitting on the very threshold that you activate a little bit, you will get exponential shortening. So based on this, and based on the counter-realization that actually there's very little evidence that KTP is ever activated under physiologic conditions in the heart, we've been stuck with the question of, well, why is it there? Why is it there at such a high density? And when does it come on? It must come on at some point, otherwise, evolutionarily, it would have probably disappeared. So to try to answer this question, 10 years ago, Bo Costa made a set of transgenes in which he made mutations of the KR6.2 subunit designed to reduce the sensitivity to ATP so that instead of it being half inhibited at 10 micromolar, the channel would be half inhibited at much higher ATP concentrations. And he made one mutation was to delete a part of the N-terminus of KR6.2, which now, based on crystal structures, we know is over here somewhere. That is enough to shift the ATP sensitivity tenfold to the right. Oh, it's not even on here. And then an additional mutation. Why isn't it on here? I'm sorry, this is the wrong. This is not the right side. An additional mutation of what we now know to be in the ATP binding site, K185Q, shifts the ATP sensitivity a further tenfold to the right, so that now we get channels that in recombinant expression are about 100-fold less sensitive to ATP than wild type. So this gives such a shift in ATP sensitivity that we would predict, any of us would predict, Yoram for sure would predict, that if you put channels into the heart that are 100-fold less sensitive to ATP, they would be active under physiologic conditions sufficient to have an effect on the action potential. Well, the striking thing was, as Catherine showed for us the first time, with telemetric ECG recordings on transgenic animals that express those transgenes, the electrocardiogram is essentially unaltered. Okay, the electrocardiogram is unaltered. There is a slight reduction of heart rate, about 15% on average. We know that the transgenes are expressed at high levels because they were GFP tagged. In fact, these were probably some of the first transgenes GFP tagged. You can see this sarcomeric pattern of expression of the transgene. By the way, it doesn't co-localize with mitotracker red, which tracks these longitudinal mitochondria, and there's no co-assembly with them. We'll come back to this point later on. But the transgenes were in the sarcolemma. So what's shown here on the left are recordings from inside-out membrane patches from wild-type ventricle. Patch was excised here at the arrow, and you see the opening of KTP channels. This is up with K on both sides of the membrane, inhibited by 5 millimolar ATP, strongly inhibited by half a millimolar ATP, and 50 micromolar in this case is around half maximal inhibition. It's probably around 30 micromolar is K1 half. But this is a recording from one of the transgenic myocytes. You see that, again, there's essentially no channel activity when the patch is on the cell. Pull it off and you see the KTP channels open. 5 millimolar now doesn't completely inhibit the channel. 50 micromolar has no effect. Half a millimolar gives very little inhibition. And in fact, half inhibition of these channels is around 2 millimolar. So it really does give about a 100-fold shift in the ATP sensitivity of the channel in the highest level expressors. Here's the ATP sensitivity recordings from those two patches above. You see about a 100-fold shift to the right. And in fact, if we looked at lots of patches from lots of cells, in white here is all the patches from wild type. You see typically half inhibition around 30 micromolar and about 300 picoamps in the patch. Here's all the ones from the high expressors. It's about 1 or 2 millimolar, but the KATP conductance is actually reduced about 5 or 10-fold. Now that in itself means that you get less channel activity, but that's not enough to explain why there's still no effect on the ECG. And as we've seen, I don't know if it's in here, yes, no effect on the action potential. So Tom Flagg then came and did a lot of work to study what's going on. He showed, this is with surface action potentials measured using suction electrodes. This was done with Anatoly Lopatin up in Michigan, from control and high-expressing transgenic myocytes. You see essentially no difference in the action potentials, although the sharp-eyed may notice a very slight difference between these two, and that is that there's a bit of an inflection in these line 4s and a little more of a dome. And in fact, we do see that these are action potentials from isolated cells. You still see a little bit more of a dome out here in the line 4. So we'll come back to why that is. Actually, maybe we won't. Maybe it's not in here. The answer is because the calcium current, it turns out, is pre-excited for some reason in these. I'm not going. There is a slight increase of calcium current in these myocytes for reasons that we really can't explain. Okay, so we said, what I've told you is that we have these transgenic mice which are expressing these ATP-insensitive channels which are very ATP-insensitive and which should be turned on, yet the ECG is not altered and the action potential is not shortened. So why not? We know, I'm not going to show you the data, but we have a long story in the pancreas that if we make channels even slightly overactive, we get neonatal diabetes, it stops insulin secretion, it's a very dramatic effect. It's actually what causes neonatal diabetes in humans. It's very striking. So what's the difference? Why doesn't this do something in the heart? Well, I told you that the dogma is that KR6.2 plus SuR1 makes the pancreas channel, this makes the cardiac channel. And one of the things we know is that the ability of ADP to activate the channel is not as effective in this combination as it is in this one. We also know from other kinds of, from hyperinsulinism mutations, that it is in large part the ability of ADP to activate the channel that is really what's relevant for activating channels physiologically in the pancreas. So you see here that when we add ADP on top of ATP, you get a very rapid and quite marked activation of the channels in SuR1. With SuR2A, it's much slower and not as effective. And there's about almost an order of magnitude shift in the relative effectiveness of ADP to stimulate SuR1. So this led us to think, well, maybe what's happening then? In the heart, it's SuR2A that's the relevant isoform, and that's what's needed to turn the channels on, so that even though we have low ATP sensitivity, as I explained at the beginning, you have to have ADP shifting the ATP sensitivity to get the channels to turn on. So maybe it's because we're not getting the ADP turn-on effect. So how can we agenda that? Let's put SuR1 in the heart. So Tom set out to make a series of transgenes in which he expressed either SuR1 or SuR2A as a control, overexpressed in the heart to put in the appropriate SuR property. So he made a whole series of transgenes, and they were flag tagged. And he made a whole series of transgenes that overexpressed at varying levels SuR1 by RNA levels, and they corresponded to protein levels. And this is 180 kilodaltons is about where we see the primary SuR1 band, and you can see that the protein levels are matching the RNA levels in the heart. That's the flag bit. But turns out, first of all, there's still no major detrimental effect, and SuR1 expression also, by the way, exerts a dominant negative effect. So 697 is one of the weaker expressing lines, 720 is one of the stronger expressing lines of SuR1, and you see that you get less KATP conductance, whether measured by whole cell or excised patch, as you raise the level of the transgene. So why is it, we could ask the question, why is it, first of all, and this is a bit of an artifact of the method, but why does overexpressing SuR1 or, as I showed earlier, overexpressing Ka6.2 subunits, either one of them has a dominant negative effect? And we suggested that it's probably stoichiometric mismatch that is responsible. So we know from the work many people, but championed a lot by Lily Jan and co, that SuR1 and Ka6.2, each of them bears a RKR motif, which is a probably fairly non-specific polycationic ER retention signal. And that each isoform expressed on its own is retained in the ER and does not effectively make it to the surface. But it appears that each of these RKR signals on either subunit is masked by the presence of the other subunit, which is in part probably why we have the stoichiometric requirement that we see. So then you get chance on the surface. Okay, so what's the obvious experiment? Cross the SUR overexpressors with the Ka6 overexpressors, then we'll get lots of overexpression of both. We'll restore the correct stoichiometry between the two subunits. We'll get lots of channels. So we did that, and in fact what we got was, what am I doing there? We got interesting, lethal phenotypes that didn't match the predictions. So what I told you is we have SUR1, and this is kind of an important technical point I want to make, that we have SUR1 expressed at different levels, and I want to concentrate on two lines, the so-called 720 line, in which it's expressed at a high level, 697, it's expressed at a much lower level. For SUR2A, we also had one high line and several that were not detectably measured, but we had one SUR2A line that was high, and by the way, this, and as with the SUR1, this SUR2A line had no major overt phenotype. Now when we crossed the lines together, we crossed them against our KR6 lines. We crossed the SUR1 720, which is the high expressing line, with either line, was line 720? With either a line 2, which is one of the lower expressing lines of KR6.2, or with so-called line 4, which is a high expressing line 4. When we crossed SUR1 720 with line 4, we never saw any live births that were double transgenic. We never genotyped a double transgene, which suggests that it's in utero lethal. When we crossed SUR1 720 with the low expressing 6.2 line, we actually had a variable onset sudden death, and about 50% of them were dead by about 8 weeks after weaning. Conversely, if we took SUR1 697, which is the low expressing SUR1 line, and crossed that with line 2, which is the low expressing Ka6 line, we got really no lethality. But if we crossed this low line with the high Ka6 line, we got another sudden death phenotype at about 8 weeks after weaning. So the point is, if we cross the high of one with the low of the other, or the low of one with the high of the other, these are independent lines where the transgene's been inserted at different points, we can get the same sudden death phenotype. So what it's telling you, or telling us, is that it is the combination of SUR1 with Ka6 that's doing it, and it has nothing to do with where the transgene's been inserted. As an important control, SUR2A, which is the supposed cardiac isoform, expressed at high level, as detected by RNA levels and protein levels, crossed with the highest level of Ka6.2, has no lethality.
So the blue data, the SUR1 720 crossed with line 2, seems to actually, they have mortality reaching 50% very much earlier.
I agree. So we haven't actually gone back and done a lot more of these. These were not done as many. So it's conceivable that this is still just the randomness of not doing so many. This is the one that Tom focused on, and with these he got a clear sort of monophasic fall. It's probably, at this juncture, and by the time we come to the end, it may be worth thinking about this, to go to look at these in more detail, and it may turn out that there is an early phenotype and then they're stable. But for now, so good point. For now, the main point to note is that SuR2A does nothing, but SuR1, when it's with KL6.2, can be lethal. And we had all kinds of arrhythmias. First of all, I just want to note that in the flag SuR1s alone, that is without the mutant KL6.2 being expressed, what we saw was a PR prolongation that was proportional to the level of expression of the transgene. So the PR interval is one of the few measures in the mouse that are quite reliable, and the delta N, KL6.2 delta N K185Q transgenes were normal, as was the SuR2A overexpressors, but the SuR1 overexpressors had quite substantial prolongation of the PR interval, particularly in the high expressors. And in the double mutants, we saw many arrhythmias, which included 2-to-1 AV block, in this case, intermittent ventricular tachycardia here, combined with AV block. This is probably atrial fibrillation, and this is sustained VT. And these arrhythmias just generally seem to get worse as they progress towards death, but we never really figured out exactly why they died. But we never saw any arrhythmias in the SuR2A double transgenics. So this didn't give us a complete explanation, but what it said was that SuR1 can be detrimental in the heart and suggests a specific role, potentially, for SuR1, if it's there. Oh, and the other point I need to make is that when we went back to look under the lamppost, we are drinkers in the lab, and we often lose our keys, and you all know, do you know the adage about the drunkard who's lost his keys? Looking under the lamppost. Did you lose your keys? Yes. Where did you lose them? I lost them over there somewhere. Well, why are you looking here? Because it's pitch black over there. So we are always looking in the ventricle, and everybody's always looking in the ventricle at ventricular myocytes who looks at these kinds of mice, because they're easy to get. And when we looked at the ventricle, where we expected to regenerate with these kinds of crosses, KATP channels with these unique properties, we got an even stronger suppression of ventricular KATP in both SuR1 and SuR2A double transgenics. Now, so that's essentially a phenotype of a KATP knockout. Well, KATP knockouts do not do this. KATP knockout animals are essentially normal in the heart. They have no KATP so that they don't respond to ischemia in the same way, but under normal conditions, they're fine. So this lethality is not because of suppression of ventricular KATP. So then we thought, well, you know, maybe SuR1 is there normally. Now, the transgenes that we're making are driven by the alpha MHC promoters, so they're expressed everywhere, and we can show that they're everywhere by green fluorescence. So we wondered maybe what we're doing with these is putting active KATP into some very specific place that is having all these detrimental consequences. And maybe SuR1 is there already. And there's reasons to think this, because there's been many bits of evidence over the years to argue that SuR1 might actually be there. So there is many reports showing that SuR1 mRNA or protein are expressed in the heart. All of these have the potential caveat that it's not myocyte tissue that's being looked at, or that the reagents are not specific enough. SuR1 antisense can inhibit KATP in cultured neonatal myocytes. And this may be more to the point that there is some evidence from atrial and ventricular KATP currents in neonatal cells that they have differential sensitivities to diazoxide. Now, diazoxide is very SuR1 specific. This is the work of a group led by Alex Berchi in Switzerland. And I should go back to this one slide that I jumped over, which shows that one property of this is beta cell-like KL6.1 channels are activated by diazoxide at very low micromolar levels, whereas cardiac KATP channels, cardiac-like SuR2 based KATP channels are essentially insensitive to diazoxide. So diazoxide is a drug that binds somewhere in the transmembrane domains and activates SuR1 but not SuR2. So I'll jump back to where I was. So, and then finally, what we published last year was that SuR1 animals, SuR1 knockout animals, which we had and that are to look at, we had them initially to look at the pancreatic phenotype, but we looked at these animals under left coronary artery bypass, left coronary occlusion with John Elrod was the first author, and the senior author was David Leffer from New York. And what we showed was that if you knock out SuR1, the animals tolerate occlusion much better than wild type, which was a kind of surprising finding. But it did suggest to us that SuR1 must be doing something in the heart so that knocking it out can do something. So we set out to make new antibodies for SuR1 because commercial antibodies for SuR1, SuR2, and the KR6 isoforms have been shown to be very non-specific and have led to a lot of results that are probably questionable. So we made a new SuR1 antibody which detects SuR1 at both the core glycosylated and the higher order glycosylated isoforms as effectively as a flag antibody does against the flag SuR1. This shows that the SuR1 has been fully processed. This higher band is not seen typically if we don't co-express KR6 with it. It's specific for SuR1. It doesn't detect SuR2 or SuR2A or 2B, even at high exposures. And what it shows us is that SuR1 is definitely present in the mouse heart. So with normal pico sensitivity detection, here is the full gel from a wild type heart with a band. The predominant band seen is at 150 under these conditions, and there is no such band in the SuR knockout, but this artifactual band at 50, around 50 kD is still there. If we take transgenic hearts that have been overexpressing SuR1, this is from line 720. If we and dilute the protein 100-fold, we get the same band showing up. At full concentration, we see the main band at 150, but now we see a whole series of additional bands at higher molecular weights, as well as one at 100 that seems to be specific. Because if we then take the wild type heart, that's this tissue, and look at it with high sensitivity femto detection, now we see all the same bands in the wild type heart that we see overexpressed in the transgenic. So this is kind of interesting because this tells us that all of these additional bands that we're seeing in our transgenic overexpressors are the same bands that we get processed from the wild type gene, just everything is much higher level. But if we take the SuR1 knockouts now with this high sensitivity femto detection, we don't see any of those bands except for one tiny little band here that might actually be a bleed over from another lane. Otherwise, there is some artifact that is very weakly detectable that happens to be at the same molecular weight. So where is it? It's in the heart. If we take isolated ventricle and atrium, we see what may be a very faint band in wild type ventricle that's gone from the SuR1 knockout, but we see a very clear core glycosylated and higher order glycosylated bands in the atrium. And in fact, they are high enough to detect them in staining. So this shows the background staining from SuR1 knockout atrium stained with the anti-SuR1 antibody, and this is from wild type. You see much higher specific staining. We see specific staining of atrial myocytes. We don't see any staining of knockouts. What you're seeing here is a white line that's been drawn around the edge of the myocytes so you can see them. In the ventricle, we really don't detect any staining above background in the wild type, suggesting that really there is no SuR1 in the wild type ventricle. And these are from the transgenics showing massive overexpression. So what this points to is that we've definitely got SuR1 in the atrial myocytes, probably not in ventricular myocytes. And then what we've shown is that in fact SuR1 is essential for the atrial KATP. This shows properties of from ventricular myocytes. This is inside-out membrane patches, wild type and SuR1 knockout with inhibition by ATP, showing the same properties for the KATP channels and more or less the same density in patches. And by whole cell patch clamp, the currents that are activated by metabolic inhibition in wild type and SuR1 knockout look the same. So there's no change in ventricular myocyte KATP properties, but the atrial KATP channels disappear in the knockout. So these are from atrial myocytes. These are KATP channels showing inhibition by ATP. SuR1 knockout, there is no detectable channels, and there is no detectable current on average. And interestingly, and this is where we come back now to what might be happening physiologically. If we do whole cell voltage clamp, and Jean says that she sees exactly the same thing, if we do whole cell voltage clamp on ventricular myocytes, we never see spontaneous activation of KATP, no matter what ATP we have in the pipette. We have to put on metabolic inhibition or pinacidil to turn the channels on. But in atrial myocytes, and unfortunately poor old Heischer, wherever she is now struggling with this. Doesn't seem to matter what ATP she puts in the pipette. In some myocytes, there is spontaneous activation of KATP, and it spontaneously activates always if there's very low or no ATP in the pipette. Now, these atrial myocytes are slightly smaller than ventricular myocytes, so they may be better dialyzed, but I don't think that's enough to explain the fact that we can frequently see the spontaneous activation of maximal KATP from atrial myocytes just by dialysis. By contrast, in SuR1 knockout myocytes, there is no KATP gets turned on. So SuR1 is actually essential for, and this is more of the same. Sometimes with a control that contains ATP, we can add diazoxide and turn the channels on. There is no such thing in the atrium. So SuR1 is essential, based on this, for the atrial KATP. And I won't bore you with this. This is just showing that the pharmacologic profile of what you see in wild-type atrium and wild-type ventricle matches SuR1 in one case, SuR2A in the other. So what's the importance of all this? Well, we're starting to address this by experiments at the cellular level and now also, more importantly, perhaps at the heart level with Alexei and with Igor and Vadim, and using their beautiful technique to actually monitor action potentials simultaneously from the atrium and ventricle of the isolated heart that's being perfused through the coronary arteries, and to monitor action potentials in the presence of blebistatin to stop contraction from both chambers. And what we can show is that in the same heart, drugs which activate SuR1 will act on the atrium, not the ventricle. Those on the ventricle will not act on the atrium. And so what this means is that in the same heart, the conditions that activate the channel, one channel, will shorten the action potential in one chamber, but not the other. So here's an example of that. In this particular case, adding diazoxide gives a shortening of the atrial action potential from 32 to 24 milliseconds, but no effect, or even a slight lengthening actually, on the ventricle, whereas then the addition of pinacidil has no effect on the atrial action potential. In fact, it gets slightly longer than control, whereas the ventricular action potential is almost halved. And this is summary data showing this effect. So a specific effect of diazoxide on the atrium, and no effect on the ventricle, and a specific effect of pinacidil on the ventricles, and no effect on the atrium. And if we take SuR1 knockout animals, we completely lose the diazoxide effect in the atrium, no effect on the pinacidil effect in the ventricle. So it's very clean in these mouse tissues. So this has important implications, which I'll spend the last couple of minutes telling you about. And I'll just show you one other piece of more preliminary data from the dog, which was done by, I'm not quite sure, I guess somebody in Charlie Ancelovich's lab, where they basically put on diazoxide in the atrium of the dog, and they showed that in fact they get nice, very nice action potential shortening in the atrium. And in fact, in the presence of diazoxide, and this is of interest to those who are interested in the potential role of KATP in fibrillation, under controlled conditions, extra stimuli could induce only one delayed beat, but in the presence of diazoxide, an extra stimulus could lead to atrial tachycardia. In the ventricle, the addition of diazoxide in endo and epicardium and visible on the ECG cause substantial atrial potential shortening, again reversible by glibenclamide. And again, in the ventricle now, in the presence of diazoxide, extra stimuli could trigger runs of ventricular tachycardia. Now this makes this finding of SuR1 in the heart potentially even more important. This suggests, doesn't prove, because diazoxide could be acting on SuR2A, I suppose, if the conditions were right, but we don't really see any activation of SuR2A by diazoxide. So more likely, the interpretation, I think, is that SuR1 is probably in the ventricle of the larger dog, and Vadim has seen some effects of diazoxide in the we won't record this bit. Vadim has seen effects of diazoxide to shorten the accidental in the ventricle as well, in human. So, yeah, actually it's in the nutshell. So it's
even bigger than the normal.
Yeah. So, in bigger animals, SuR1 may be even more important in the ventricle. So this whole idea that that there is what are the implications? So what? So this whole idea that SuR isoforms are specific between the pancreas and the heart, and that all of the drug effects that we see for the pancreas will not apply to the heart is probably not true, and that therefore the diazoxide may be acting on the heart. So one of the cases where, and further implications, I can't remember what order I have these in, but one major potential implication of what we're seeing is the pharmacology of what's called ischemic preconditioning. This is something which has received a lot of interest from sort of more clinically applied researchers. The general idea is that if you precondition hearts to ischemia, you give a brief period of ischemia. If the timing is right, then the heart is much better able to survive a longer period of ischemia with less damage. So there is a lot of searching around for preconditioning mimetic drugs or mechanisms to induce this so-called preconditioning phenomenon that leads to better recovery afterwards. Now, one of the findings is that ischemic preconditioning is mimicked by diazoxide. And this has always been understood that because diazoxide doesn't work on sarcolemmal KATP, it must work on something else. And there is this long-standing notion, for which there is little experimental evidence, of the existence of a mitochondrial KATP. Almost certainly none of the isoforms I've talked about are present in the mitochondria, so that's not mito-KATP. And the biophysical evidence for a K channel that is inhibited by ATP is also minimal. However, there is clear evidence that diazoxide does something to mitochondria. Probably it's acting on another protein. But the implication has always been that it works on mitochondrial KATP channels, and so if you do a PubMed search for cardiac ischemic preconditioning, I found 2,500 papers on it, and for diazoxide preconditioning in the heart, 216. All of these are based on simplistic pharmacology. You just put on diazoxide, you see an effect, and they all interpret it as mitochondrial KATP function. Perhaps it isn't. Perhaps it's an effect of diazoxide on the sarcolemmal KATP channel affecting the electrophysiologic substrate and thereby affecting what happens during ischemia, possibly causing action potential shortening, which in and of itself will reduce the calcium loading during the ischemic period. You turn on KATP, this is the old understanding of what's the role of KATP in the first place. It shortens the action potential, it stops calcium entry, so at a cell level it's protective in terms of reducing the potential damage from calcium overload. Maybe this is really what's happening in preconditioning. And in fact, if you go back to KATP knockout animals, it's shown that not only does is KATP necessary for ischemic protection, but it's also necessary for preconditioning. So ischemic protection, here's an example from KATP wild type hearts. Ischemia is induced at this point. This is a Langendorf perfused heart. The contraction stops very quickly, and then the heart stays there and then gradually begins to go into rigor at the end of the ischemic period, but it's not in complete rigor. And then when reperfusion is restored, there's some arrhythmias, maybe bad, and then gradually contractility comes back again. But the general finding was that in knockout and K6.2 knockout animals, when ischemia is induced, actually contraction doesn't stop for much longer. It goes on, and in fact it's still going on probably at the beginning of rigor. And this is what I showed this a long, long time ago in myocytes too, that if you actually voltage clamp myocytes that are metabolically inhibited, you can keep contraction going all the way into rigor until the whole thing seizes up. So what this means is that at this point, at the end of this ischemic period, this heart is probably much more calcium loaded than this one. In fact, you can see that rigor is plateaued at this point. And now when the flow is restored, there is no recovery of contraction. Now what happens in preconditioning, this is the protocol that was used to look at preconditioning in wild type and knockout hearts. And this is wild type control, wild type preconditioning. You see this great reduction of infarct size. In the knockout animals, there's no effect of this preconditioning protocol on infarct size. And when this was published by Marban and Sano and co, there was some lines in there about how, well, this is perhaps because mice have small hearts, and some notion that small hearts and high heart rates means that we can ignore the fact that knocking out the KATP channel gets rid of preconditioning, because in big animals it can't have anything to do with sarcolemmal KATP. There's a lot of dogma involved in all that statement. And frankly, I don't really believe it. I suspect that if you knocked out the KATP in humans, you would also lose preconditioning. And I think that it may have a lot to do with the fact, or the likelihood, that the sarcolemmal channel is also involved in preconditioning, although at this juncture we don't really know how.
So do people who are on glutamate and those sorts of things have worse outcomes?
That's still some issue. There are some reports that they could be. It's very difficult to control properly, because in people who are on glutamate, they are diabetic and they have all the complications associated with diabetes. So where do we go from here? We want to look at preconditioning in SuR1 knockout hearts and see whether that really affects preconditioning. Potentially there could be loss of specific diazoxide-induced preconditioning. We'd also like to look at mitochondria and the mitochondrial function in SuR1 knockout animals, and we may do that in collaboration with Jennifer Lawton surgery. Obviously, we'd like to look at diazoxide effects in human hearts, and this is really what Vadim and co are doing. And ultimately, we'd like to look at KATP mutations in people. We actually have a family who have got neonatal diabetes, and the short answer is that we have put a Holter monitor on a woman who was a neonatal diabetic for 25 years. She's been on insulin all this time, and while she was on insulin, she was Holter monitored. She also did a stress test, and we still didn't see anything there. And this is a woman who has neonatal diabetes because of a gain of function in the KATP channel. She's basically K6.2, like our delta N K to Q gain of function, and that's what caused her neonatal diabetes. Under those conditions, her ECG looks more or less normal, although she has quite substantial heart rate variability that may be out of the normal range. She is now on sulfonylureas, which should have blocked the channels, and we want to bring her back in. We don't have her yet to look at Holter monitor. But I think in the long run, we'd like to look at more of these people, and particularly what are now we're getting, well, we don't have any here in St. Louis, but we're in connection with some in various parts of the world, who actually have gain of function in SuR1 that is causing their neonatal diabetes. So they actually have a gain of function in SuR1. So that, I think, could well be doing something in the heart. So I think I acknowledge most of the people who did most of the work. Bo generated the animals in the first place for the first sets of transgenes. Tom did most of the characterization. The technicians in the lab did a lot towards taking care of the animals. Ricard did a lot of our work on comparing SuR1 to SuR2, and then a whole host of critical collaborators did much of the experiments that I showed you. Maria was involved in a lot of the late experiments with Tom, particularly Catherine, Anatoly, Mark, Helena, and now Igor, Vadim, and Alexei for the whole heart studies. Thanks a lot.
Thank you, Colin, for an excellent talk. Any questions?
Can you try this antibody on immunohistochemistry in humans?
Yes, we can. And so today we were late coming over here because Haisha was killing SuR1 and wild-type animals and isolating the hearts and perfusing them and then freezing them. And they went in a FedEx box tonight to go to Helena in Manchester, who will section them to look at the conducting tissue in mice. And it's probably a good idea to wait and see what we get there. But as the human tissues come available, we should try that. We have more to do to confirm, I guess, that the antibodies work in humans. They would, I mean, the sequence of the SuR1 is very conserved, and I'm pretty sure that the sequence to which the antibody was generated is common to humans, so it should work.
Colin, does PNCD also affect on mitochondria and KATP channels?
I'm sorry.
I'm talking about PNCD.
No, just say it again. I'm sorry, my ears are
Does
popping.
PNCD also affect on mitochondria like a diazoxide?
I think not, actually.
Uh-huh.
I think not. There is also this issue of 5-hydroxydecanoate is supposed to block the mitochondrial channels, and I was kind of hoping we might have enough data to show you, but we don't really yet. On atrial myocytes, Haisha has got some data to suggest that 5-HD can slow or retard the activation of the sarcolemmal KATP, but it doesn't block it in the long run. But 5-hydroxydecanoate is a fatty acid, so this is not a small molecule blocker of the channel. So how it works, I've really no idea, but we're going to try and look at that in more detail. I suspect that there really is something going on in the mitochondria, although I don't think it's through any KATP channel.
Are the SuR1 channels in the heart inactivated by sulfonylureas, just like the SuR1 channels in the brain?
Inhibited, yes.
So that's known, they respond similarly.
Mm-hmm. Yeah, so then you could say, well, how come all these diabetics aren't affected by giving sulfonylureas? Why doesn't it do something bad in their heart?
But they may.
They may, and
there's a very large, frequently suppressed body of information from the University Group Diabetes Program 30 years ago, where people who took sulfonylureas were more likely to experience subdip. And that's just been lost, from currently clinically conversations. So,
NYU no longer prescribes sulfonylureas as their first-line type 2 diabetic agents because of that.
because of that effect, not just because metformin's better. Well,
there's a substantial history about that here in Washington University because people at the medical school believe very fervently in these, and then subpoenaed the people who did the studies, went through all the data, and there was a concentration of deaths at one center. And so that seemed to diffuse the concern that it was a big deal. But there's always been a very real worry that sulfonylureas may kill people, and there's an impression of that in clinical practice, and yet they've been around forever.
there is an impression in clinical practice that this can happen, that you can get sudden deaths on it. Yeah, I mean, I'm quite happy to believe it. I mean, the dogma answer to why it's not a problem is, oh, well, because KATP is not activated. But maybe it is some. I mean, it must be sometimes. I mean, just if you do one of those back of the envelope calculations, each KATP channel is a megadalton complex. It's a huge protein complex, and we've never found any evidence for it doing anything except being a KATP channel. And it is the most dense K channel in the heart, in the myocardium, in ventricle and atrium by a long shot. It is also more dense, or as dense essentially, as the sodium channel. So if you turn them on, you can stop the heart. I mean, there has been sort of preclinical efforts to use KATP activation to stop the heart for surgery. So it has to stay closed mostly, but with such a huge density, anything that begins to turn it on will do something. So I agree.
How does the anatomy of these knockouts or overexpressed looks? Is there any relation with hypertrophy? You're thinking of that?
So in the knockouts, there's no evidence for any morphologic change in the heart. In the transgenics, many of them are normal, but we do see atrial hypertrophy in some cases in the SuR1 double transgenics. So some of those arrhythmias, there's some atrial hypertrophy, which we've never fully characterized it, but so at this point our guess is, and this is what Aisha is now working hard to show, that what's happening in those double transgenics, or in the SuR1 transgenics in the first place, is that it's not initially at least a ventricular phenomenon. It is something to do with altering supraventricular substrates, either atrial muscle or in the pacemaking or conducting tissue somewhere. That overactive KATP is what then is leading downstream to all these other effects, which may relate to Igor's point about how it's possible that in one of those paired lines we've got evidence that we get early death and then it's lost. So it may be that it kills them early on or they get through it.
So actually I have a comment. I've seen several pieces of evidence that KATP plays a major role in cardiac protection in hibernating species. Actually some piece of evidence I've seen which was not yet published, but they suggested that KATP is expressed in mitochondria and specifically perhaps at larger quantities during hibernation season. But what I'm trying to say is that what is known about connexin 43, it's also overexpressed during hibernating season, and Rob Gordy has a theory that connexin 43 is one of the proteins which clearly helps a lot during hibernation, and there is well, because essentially cell-cell coupling is improved by overexpression of connexin 43 during low temperature, and it maintains conduction. So have safe conduction even at temperatures as 1 degree centigrade, even at 0 degree centigrade.
So it has nothing to do with changing the rate,
just pure cell-cell coupling. But what was puzzling in general, why mammals have so much of connexin 43 as compared to, let's say, birds which don't have connexin 43 at all. And again, Rob Gordy suggested that perhaps the reason is we all are descendants of hibernators. So early mammals were hibernators. And that's why we inherited this large quantity of connexin 43. And if we would have a chance to hibernate, maybe we would also overexpress it. So KATP perhaps is a similar.
KATP is in birds.
Well, but overexpression and a large quantity in mammals perhaps is a remnant.
It's at high quantity in what I've looked at myself. Frog ventricle has a lot of KATP, so maybe not. Good idea, and I guess frogs are descendants of birds, so.
Colin, do you or does anyone know about expression of MHC?
Frogs could get really cold, couldn't they? Yeah. Sorry, go on.
Yeah. MHC expression in the conduction system. You mentioned the conduction system, so I'm just curious about where a transgenic overexpressor is really overexpressing.
Yeah, I don't have them in here, but it's everywhere. So Helena did section out transgenics, and there is the delta and K to Q GFP tag transgenics. That's everywhere.
So if you look specifically at the conduction system and say the His bundle.
Well, she did sections through the SA node and sections through the AV node, not the His bundle per se. But it's in the SA node and AV node, the MHC driven transgene. That's all I really know. But I'm going to guess it's in there.
Is there also SuR2 in the canine ventricle, and do these things form heteromeric channels between 6.2 and different SuR? That that would have different pharmacological properties?
So there's several questions there. So one, I'm sure that SuR2A is also in the canine ventricle because I'm certain that there is panacetyl action in the canine ventricle. The question about whether these associate into heteromeric channels is unanswered in native tissues, but there is, we and another group just published last year demonstrating in recombinant channels with appropriate tags and labeling that we can do it, that shows that you can get SuR1 and SuR2 into the same channel complex. So channels that have both subunits in there. And what you get is mixed pharmacology between the two. I mean, you know, you see both. So you get a kind of averaging of the two subunits.
That's only expression system.
In a recombinant expression system, yeah. So in native cells, who knows? I mean, you know, going back to the original gene structure of these things, it's really fascinating that two downstream genes of one another in two pairs, and you can get each. So I'm sure it all has to do with common regulatory elements that are detectable in each combo. But nobody's really looked at this. I think Tom Flagg's going to try and do it.
So, what about SuR2B? So expressed in vascular cells. Is there some pharmacological agent to selectively, specifically regulate it?
Actually, SuR2B, I think, it does have a differential sensitivity that it is sensitive both, I think, to panacetyl and diazoxide. So,
but there is no agent which only affects.
No.
Okay, any other? All right, thanks again.
Thank
We can all go hibernate.
Channels. And of course, I don't think I need sulfonylurea receptor with the two nucleotide binding folds, which the electrocardiogram is essentially unaltered. These were probably some of the first transgenes GFP tag. You can see with this anti-SuR1 antibody, and this is from wild type, you see much. The ventricle, we really don't detect any staining above background. So I think I acknowledge most of the people who did most of the work below. A lot of our work on comparing SuR1 to SuR2. Thanks a lot. Yes, we can. And so today we have a FedEx box tonight to go to Helena in Manchester. We have more to do to confirm, I guess, that the antibodies work in humans. They would work. I think not, actually. I think on atrial myocytes, HiSuR has shown 5-hydroxydecanoate is a fatty acid. So, you know, this is not a KATP channel. Mm-hmm. Yeah, so then you could say, well, how come all these diabetics?
You know, from currently clinically conversation, well, we did the studies, because we went through all the data, and there was a
I mean, you know, the dogma answer to why it's not a problem. The megadalton complex. It's a huge protein complex, and we've never found any. There has been sort of preclinical efforts to do something. So I agree. Any morphologic change in the heart, HiSuR is now working hard to show that what's happening supraventricular substrates, either atrial. It's possible that in one of those paired lines we've got evidence.
Actually, I have a comment. I've seen several but they suggested that KATP is expressed helps a lot during hibernation and pure cell coupling. But what was puzzling in general, why mammals have hibernators? And that's why we inherited this large quantity of connexin. Large quantity in mammals perhaps is a remnant.
It's at high quality, so.
Colin, do you or does anyone?
Helena did section out transgenics, and there is SA node and AV node. The MHC-driven transgenic.
Channels between 6.6
into heteromeric channels is unanswered in native tissues, and labeling that we can do it that shows the homology between the two. I mean, you know, you see both in two pairs, and you can get each. So I'm sure it all has to.
Pharmacological agent to
It's
selectively, specifically.